sound occurs because you get these compressions and rarefactions in the air molecules. So those molecules are compressing against one another, and then they're rarefying. They're spreading out, and it's not like one molecule is moving all the way down. It's just the pressure wave that is moving down. Okay. Okay. Let's talk about how we describe an auditory stimulus. We have these sinusoidal changes in pressure. That's one way to do it. And we're going to think about the parameters of frequency, amplitude, and phase. We'll also look at period and wavelength, but most of the time we describe things in terms of frequency, amplitude, and phase. Okay? Amplitude, right, if we're talking about pressure intensity, it's just the distance between this resting point and the loudest point. So amplitude is what you think it is, right? Relates to intensity, right? The psychological percept of that would be what? Loudness. Yeah, loud. All right. Frequency. Okay, the number of cycles per second, which is the unit for that is hertz. Okay, so if this is 1 second, okay, I've gone through 2 cycles, right? So that's a 2 hertz signal. Right, so if I know how much I've got over time, I can tell what the frequency is. Okay. All right. All right. Okay. So long wavelengths are low frequency, and short wavelengths are high frequency. Okay. A wavelength is the distance from the start of a cycle till you get to the next start of the cycle. So if we're talking about in this case it's not distance, but if we were talking about distance, that would be your wavelength. So just remember that low frequencies have long wavelengths, and high frequencies have short wavelengths. Period is just 1 over frequency. So if I can determine what the period is if I know what the frequency is over what period of time. So we're not going to really spend much time on period. Okay? Let's keep going. Wavelength, as I said, is the distance between similar points in the waveform. So if this is over space, from here till the time I reach that point again in the waveform is the wavelength. Okay? Long wavelengths are low frequency, short wavelengths are high frequency. And that's important for how the auditory system deals with that information, right?
Can you repeat that one more time? What?
Sure. So long wavelengths are low frequency. Long, low. Easy way to remember it. Okay, and short wavelengths are high frequency, or high frequencies have short wavelengths. Okay? Remember when we talked about sound localization and how sound gets obstructed by the head and it could get obstructed by other objects, the wavelength matters in that case. So we'll look at that in a second. Phase is just where you are in the cycle of the waveform. So these two things are about 90 degrees out of phase. Okay, so if this is 0, this is 90, 180, 270, 360. I'm sure you guys have all seen this stuff before. Okay, and we looked at it in the visual system earlier in class too. So phase is just that. And we don't deal a whole lot with phase, so we're not very sensitive to phase, unless we're talking about phase between the 2 ears. Okay, the dynamic range of the auditory system is huge, right? It's a factor of 1,000,000 from our threshold to our 120 dB, which is where we're getting into the pain region. Okay, these should all be pretty straightforward to you. The physical stimuli and the psychological correlates. Frequency is pitch, amplitude or intensity is loudness. Phase, again, we're not sensitive to that. And harmonic structure is timbre. Do you remember that? Remember timbre? We'll look at that here in a sec. Okay, we look at waveforms in 2 ways. Acoustic stimuli in 2 ways. So we've got a waveform and we've got a magnitude spectrum. So this is basically showing what the signal is doing over time. So the amplitude of that signal over time. Here, this is a sinusoid, but it could be much more complicated than that, right? And this is if we were to break that signal down into its individual frequency components, we would represent it this way. So that signal has a frequency component at 5 hertz, 10 hertz, and 15 hertz. Not that one, but if we were to do one. Okay? What do you call it when you have a complex signal and you break it down into its simple parts, its sinusoidal components? What kind of analysis is that? Fourier analysis. Okay? The ear is a Fourier analyzer. Remember how the cochlear broke a complex signal down and you saw different places along the basilar membrane where there were resonances for each frequency? So the ear performs a Fourier analysis, and that's called Ohm's law. We'll see that. We'll go over that again. All right. If we're thinking about this magnitude spectrum representation, okay? Harmonics, remember, so if this is our fundamental frequency, a harmonic is an integer multiple of that fundamental frequency. So this is 2 times f0, this is 3 times f0. All right? Each doubling of frequency is an octave. Okay? So, if this is 100 hertz, and this is 200 hertz, I've gone up 1 octave. If this is 300 hertz, I've gone up 2 octaves from this. But an octave up from 200 hertz is what? 400 hertz, right? And an octave up from 400 hertz, 800 hertz, right? So it keeps doubling, right? And because the ear analyzes sound using that kind of octave scale, remember how we saw how the auditory filters got broader and broader as we got higher and higher in frequency. We'll look at that again in a second too. That's because those octaves are getting larger and larger in terms of the number of frequencies, right? So a distance between 100 and 200 hertz on the basilar membrane may be the same as a distance between 800 and 1600 hertz on the basilar membrane. Okay, timbre, right? It's just quality of sound or the color of sound, and it's what allows you to say for a given note played by 3 different instruments, which instrument was it? Okay? So if this is a tuning fork, this is a pure tone, just a single, this would represent a single sinusoid. Okay? A single simple sinusoid at whatever frequency this is. Okay? Now we play that same note through a flute, and you get these different harmonics above that fundamental frequency. It's still going to have that pitch, but it's going to have these different harmonics, a different number of harmonics and different levels of those harmonics. Same thing here, same pitch, but the clarinet is going to be described differently by a different set of harmonics at different levels. Okay? That's timbre. Fourier analysis, we know that. Let's talk about the ear. Okay? Outer ear structures. Pinna, external auditory meatus, and tympanic membrane. Okay? If we have questions about that, I want to know the real names of them. Okay? Okay, what does the outer ear do? Well, it collects sound. Right? The outer ear can help with localization. And we'll look at that more in a few minutes. Yeah?
I don't want this to be a retarded question, but clearly that picture up there is labeled. So are we going to have to label those structures?
That may be a question on the exam. There is going to be some labeling of something. It may be the ear, it may be another sensory system, it may be a different part of the ear. Okay, so you want to know that. There's a lot in here, but it's it's pretty straightforward. So let's go through it. But remember now, we've got the outer ear, the middle ear, and the inner ear. So this is a nice picture to show you where those things are. Okay, here's what the outer ear is doing. Here are the things that it says ear canal. It's really, again, it's external auditory meatus, and the tympanic membrane is the eardrum. So these are sort of the colloquial terms, but we really want the actual terms here. All right? At least know what they are. Maybe you'll get partial credit on things. Okay? But I would like you to know those real names. Okay, outer ear functions, again, collect and amplify the sound, helps with localization, and it helps protect your eardrum because eardrum is all the way down in that ear canal, or external auditory meatus. Okay, the middle ear. Okay, so right after we get to the tympanic membrane, on the other side of the tympanic membrane is what? The ossicles, those three bones, the smallest bones in our body, the malleus, incus, and the stapes. Okay, remember how they're sort of attached? So the malleus is touching the tympanic membrane, the incus is touching the malleus, and it is also connected to the stapes. And the stapes touches on the oval window of the cochlea. So, sound's coming in, it's pushing on the tympanic membrane, it's getting those ossicles moving, and they're pushing on the oval window, and it pushes the fluid in the cochlea. All right? So, ossicles, that's the name of the bones, okay? Which are these three, malleus, incus, and stapes, or hammer, anvil, and stirrup. Again, I'd like you to use those, the actual terms. I will give partial credit if you can't get them. Eustachian tube. And then what does the middle ear do? Okay, so the middle ear has a very important role, because remember, whenever we change from one medium to the other, we're going to lose some intensity. So if we're going from air and we're trying to use air to push on something that's mechanical, that's stiffer, you need to actually, and eventually we're going to push on the fluid in the cochlea, we need a way of amplifying that sound. Okay, so that's what the middle ear is doing. It does this impedance match. When we're going from airborne sound amplified to move the cochlear fluid. I mean, think about it. If you just air pushing on water is not going to give you, there's a lot of resistance there. Okay? So we actually get some additional amplification from those inner ear bones. Okay, how do we do it? Remember, there's these two ways. There's this area reduction. Remember, that is because we're going from a very large area on the tympanic membrane to a very small area on the stapes, right? Again, if we had a whole bunch of force coming into a single point, like you might on the heel of a shoe, a pointy heel, right? That's going to be a lot more force than if you had the heel was a big platform shoe, right? And it would just be as wide as it is on the bottom as it is on the top, right? So area reduction gives you amplification, and this lever action effect, okay? Because you've got the incus longer than the stapes, you're getting this force that's actually pushing it that way. It gives you some additional amplification. So a very important function of the middle ear is the amplification. That is the most important. It also does provide protection. Remember, we have this acoustic reflex where the muscles are attached to the malleus and the stapes, and they sort of tighten up for a loud sound to keep them from pushing too hard on the cochlea. So it helps protect us. Okay so far? Keep on moving. Okay, cochlea. Cochlea is a little bit more complicated. Remember, it's got this spiral shape to it, and it's got these different canals. Okay, the scala vestibuli, the scala media, and the scala tympani. Right, and it has these membranes that actually separate the different canals. Okay. Back to that in a second. So the stapes, I'm sorry about the quality of this picture, but the stapes is pushing here on the oval window and is pushing the fluid in the cochlear duct. The fluid that force is going all the way up here to the end, okay? Now the basilar membrane is running through here, but it doesn't touch the very end. There's this little area where the fluid pressure can turn back around and come back out. That's called the helicotrema. And when the pressure comes back out, it's pushing out on the round window. So, okay, so here are your different scala with the different fluids in them. The center thing is the organ of Corti. That is your main organ of hearing. That's where transduction takes place. What is in there? That's where your hair cells are. okay? So here's the organ of Corti, and we'll definitely we'll spend more time on that than we will on those other things, but you'll need to know those other things. Okay, so the organ of Corti sits on top of the basilar membrane. Okay, when there's a change in pressure, the fluid moves, and what you're getting is a transduction in the organ of Corti from mechanical energy into neural spikes. Okay, so how does that happen? You have this membrane on top. Let me get you a better picture of that we've seen before. Okay, you've got your tectorial membrane, okay, that sits on top, and you've got the basilar membrane that the organ of Corti is sitting on. Okay, when you get the pressure from the fluid, the basilar membrane starts moving, and it moves relative to the tectorial membrane, which is actually hinged on one end. Okay, so if this is moving and this is hinged, what's going to happen is you're going to get the hair cells starting to bend, and that's called a shearing force, and it's that shearing force that that leads to transduction. Okay, let's look at for a second at the organ of Corti. Here's your inner hair cell and outer hair cells. So you have 3 rows of outer hair cells and 1 row of inner hair cells. Okay, and that's what we're showing here as well. So you see that you get these stereocilia that are actually poking up from the outer hair cells into the tectorial membrane. They're not on the inner hair cell, but the inner hair cell stereocilia are still moving. So what causes them to fire? Okay, so here's a good depiction of that motion and what leads to that shearing force. Okay, so that shearing force is bending those stereocilia at the top of the hair cells. And what happens is when it bends, some ion channels are opened up. Each of those little tips has a little link from one to the next. Okay, and they're sort of attached to these little doorways that open up. So when you actually bend the hair cells, those tip links will cause the little ion channels to open up, okay, and allow for eventually the depolarization and the action potential to have that cell fire. Okay, so that's functionally how that works. It's a good close-up of these tip links. And see, as they bend, the little doorway, if you will, opens up. It's got that there. I'm going to keep moving because we have a lot to go. If you can follow this when you're looking at it, right? I've just tried to lay it out so that we just talk about the chain of events that occurs to help you sort of think about how things are happening. Okay? Then you get the rapid cell depolarization, which opens the channels at the hair cell's base. These neurotransmitters go to the auditory nerve fibers, okay? And then those fibers are sending signals into the higher centers of the auditory system. Here's just another picture that might help you visualize that. So this is where your basilar membrane is, okay, in this scala media, middle, okay, and then you have these other scala, and so what's happening is the fluid is pushing on here, it's going around up through, here's the helicotrema, up here, so the pressure is moving up through here, that's what we care about. Basilar membrane stops, okay, and it just comes back around and out, and then you get a full pressure out through the round window. So, there are a lot more photoreceptors than there are auditory nerve fibers or hair cells, but we get superior temporal properties in the auditory system. Let's talk about how we encode some of that information. So amplitude's encoded. Remember we saw some videos of the tympanic membrane, and the more it was pushed in and out, the higher the amplitude of this, the higher the amplitude of the signal, the more we push in and out on the tympanic membrane. So that's one way we are actually transferring that information to the auditory system. Okay? And it causes greater forces all the way down the chain. The tentorial membrane, there's more shearing force, so you're getting more firing, okay? More neurotransmitters releasing, and then more firing of the auditory nerve fiber. How do we encode frequency? We talked about a couple of different ways. Place code is one of the main ways that we encode frequency. So remember, when you've got this pressure wave that's going down the auditory system, the basilar membrane is moving, okay? And you get this maximum displacement at some place along the basilar membrane. Wherever that maximum displacement is, that's this frequency that you're going to encode. So let's think about this, because these are things you're going to know, need to know. If this is your basilar membrane, here's your base, here's your apex, okay? If we look at what's encoded at the base, okay, it's a pretty high frequency, okay? And then lower and lower frequencies, and the lowest frequencies are encoded at the apex. So when you're pushing on the oval window, the very first thing, the basilar membrane is right there, the very first thing that's encoded is the high frequencies. So if it's a high frequency sound, just because of the mechanics of the system and the structure of the basilar membrane, which is thick down at the base but narrow, and thin out at the apex but wide, that mechanical structure causes it to have a maximum displacement at a certain place based on what the frequency is. So a high frequency sound is going to cause a maximum displacement near the base, and a low frequency sound is going to have maximum displacement near the apex. Right? So different places along the basilar membrane are used to encode frequencies. Okay? That wave that moves down the basilar membrane is called what? Traveling wave, yes. That's going to be a question, I bet. So think about how we encode frequency in the place theory. All right, and if we took a look, this is sort of a cartoon look at the cochlea, we see that the highest frequencies are encoded here near the base. Well, we're first going to be pushing on the oval window, and the lowest frequencies are encoded at the apex. Here's another depiction of the traveling wave. So it's not like you get just displacement at one place. That wave is going to be moving all the way down the basilar membrane, but someplace you're going to have this maximum peak in it. That's where you're going to get the most firing, and that's going to be associated with whatever frequency is coming in. Okay? So when you get the firing along the basilar membrane, okay, you've got all these hair cells that are here on the tectorial membrane, right? And then they're going to be firing and sending information to the auditory nerves. So if I've got a high frequency sound, and I'm going to get some maximum displacement near the base, and the auditory nerve fiber then is going to send information eventually to the brain, but to the higher centers first, about a high frequency signal. Okay, so again, place along the basilar membrane. And you can see here this thickness and narrowness, and then it's a thin tissue here, but it's much wider. So it's just the mechanics of that particular tissue that's causing it to have a maximum displacement at different locations along it as a function of frequency. Okay, remember we talked about the fact that we're very good at frequency discrimination, but if we looked at the tuning of these filters, if we looked at how we encode this information, it's not very precise. You get pretty broad displacement regions. Okay? So that can't really explain how good we are at encoding frequency and discriminating frequency. At least those are the measures that some of the early scientists got when they were looking at cadavers. Okay? And they were looking at, because it's just a physical structure, they could actually put a signal in and measure where the displacement is and see what the tuning is. I can put something in at 100 hertz and see where that displacement is, and then 200 hertz and see where the displacement is, and see how narrow those are, how close those are to one another. Okay? What we know happens is that those outer hair cells are actually causing the tuning to sharpen, because what happens is there are efferent fibers that are coming down from the brain after you get a signal that says, okay, this is the frequency that you're supposed to be encoding here, so that the outer hair cells around that are stiffening up to sharpen up the response of the basilar membrane in that frequency region. Remember we saw the little hair cell dancing up and down? Okay? That's the active process of the outer hair cells. And they play a very important role. So most of the frequency information is carried by the inner hair cells. Most of the information that's going up to the brain, the afferents, is on the inner hair cells. But you get some efferents coming down that are controlling this motility of the outer hair cells to sharpen up the tuning. That's outer hair cell motility is that movement. So remember motility. Okay, here's just a depiction of how these fibers are actually. So here are your outer hair cells, and here are your inner hair cells. Right. So we've got the 3 rows. Okay, and you've got a lot of these outer hair cells that are actually on a single fiber, right? Whereas you have a lot of fibers on a single inner hair cell. Okay, so that's telling us that there's a lot more information on those single inner hair cells. And so that's important information. Okay, here's our picture. Yeah, that's the picture of the hair cell that was moving up and down. All right, so that motility, that movement is actually stiffening up the region around that frequency so you get a sharper peak. Go higher up in the system. Here are some terms you need to know. Characteristic frequency. It's a frequency that increases the firing rate of a specific neuron at the lowest intensity. So, if I stuck a probe in a neuron and I played a bunch of sounds and I wanted to play that sound loud enough to get a response out of that neuron. Okay, how loud would I have to make it? Well, if I'm playing something around 1 kilohertz or 1,000 hertz, I'm going to have to make it about 60 dB for this particular neuron in order for it to fire. Okay? But if I were to play something around maybe 9 kilohertz, I only have to make it a couple of dB, maybe 5 dB to make it fire. Right? So that's saying that that neuron is most sensitive to that frequency. Right? I don't have to put very much level into the signal in order to get it to respond. That's the characteristic frequency of that neuron. And each neuron has a different characteristic frequency. And the way we hear is based on the fact that we have a whole bunch of neurons with all these different characteristic frequencies. Okay? And if we were to measure all these different neurons, the characteristic frequency of all these different neurons, we would get something that looks like this. And then if you actually extrapolate from that and you draw a curve under the response of all those neurons, what you see is something that looks like our threshold of hearing. Right? Because at each place, we're looking at the characteristic frequency, so the lowest level that'll make that neuron respond, that auditory nerve respond. Okay. This is sort of relating what we just talked about to what we talked about a few minutes ago. So here you have a low frequency tone and a higher frequency tone. And what you see is different places along the basilar membrane where you get that maximum displacement. Okay? So, what's going to be, which tone is going to be causing maximum displacement right here? High or low frequency? High. Right? High right near the base, low near the apex. So what you get is firing of the auditory nerves, which are connected to the hair cells on the basilar membrane at that lower frequency. So maximum firing here. You may get a little spurious firing around it, and sometimes spontaneous firing of those neurons, but you get maximum here. And for the lower frequency you get maximum firing here. So this is just another way of thinking about how the response of the basal membrane leads to the auditory nerve firing associated with that particular frequency. All right, we talked about rate saturation too. So remember that auditory nerve fibers tend to have the sharpest tuning for a pretty low intensity signal, right? But well, since signals aren't that low of intensity, what happens is as you go up in level, you get each auditory nerve fiber responding to more and more frequencies, right? You sort of get this spreading of energy around as you get higher and higher in level. So the best way, the sharpest tuning of an auditory nerve is seen when you've got a low level signal. But you get this range over which that auditory nerve is going to fire that increases with increasing level. Okay? So that's our saturation. Now, that's not very good. It doesn't help us with our frequency discrimination. If every auditory nerve fiber is responding to a whole bunch of frequencies, right, then we're not going to have any good discrimination. So we have to figure out a way to use the auditory nerve fibers that we have to respond to signals at low levels and high levels, because not all signals are at 20 dB. That's pretty quiet. I'm talking at a level about 65 dB now. So we've got these two different kinds of neurons: high spontaneous firing rate neurons and low spontaneous firing rate neurons. Okay? Now, the high spontaneous firing rate neurons are just that. They have a high spontaneous firing rate, so they're firing a lot. So it doesn't take a whole lot of additional signal to get them to really fire and signal the presence of a stimulus. Okay? Something beyond their spontaneous rate. Okay? High spontaneous firing. They're already firing. Let's think about the high spontaneous firing rate, a lot of spikes, is it being really sensitive and really ready for a stimulus. So just a little stimulus is going to make it go. Right? These blue lines represent different high spontaneous firing rate neurons, the responses of them. And what you see is that for a very low signal, you start to get a response, okay, and then it sort of levels off. Okay? We have also, so those neurons are going to be good for encoding quieter signals, right? Because they're sensitive to those quieter ones. Again, lots of activity means that they're like ready to fire. Okay, so it doesn't take much of a signal. These low spontaneous firing rate neurons are firing at a slower rate, and so in order to get them to really respond, you have to put in a higher level signal. Okay, so they're not quite, think about them as maybe they're not quite as ready to fire until you put in something that's more intense. So you can see here, these red lines, which represent those low spontaneous firing rate, they don't fire for a while. And then finally they start firing. Okay, it's at a much higher level than when these started firing. Okay, and then they sort of go up slowly, and they continue to sort of rise. So they don't saturate as much. They respond over a much larger range of intensities in different ways. So these will continue to respond, but it just sort of levels off. It doesn't really have any change in response. Those low spontaneous firing rate ones will continue to increase their response as they are provided with higher and higher level stimuli. Okay. All right. So we talked about how we encode frequency via place on the basilar membrane. We also encode frequency via time. There's something called phase locking, okay, and that means that the auditory nerve is going to fire to the same place in the signal each time, the same point in the phase of the signal every time. So every time this signal hits this point in the phase, which is, I don't know, let's say it's 300 or something, 300 degrees. Okay, when I get back to that point again, it's going to fire again. And I get point there again, it's going to fire again. So I've locked to the phase of the signal, phase locking. Okay, there's a little spread here, but the probability that you're going to fire is going to be highest right at that phase. If it can fire like that, it's going to tell you something about the frequency. Now, we run into a problem. Because remember that neurons can't fire at an infinite rate. About 1,000 hertz, about 1,000 times a second, is about as fast as they can fire, these auditory neurons. Okay? So we can encode higher frequency signals through phase locking. We do have another way we can encode slightly higher frequency signals, and this is through this volley principle, where neuron A fires at a particular point in the phase of the signal. Neuron B will also fire, okay, at that point in the phase of the signal, but it may have been a couple cycles later. So they're all firing essentially at the same phase of the signal as one another, but because they can't keep up with how fast it's changing with the frequency, they only sort of catch it every couple of times through. And so all these neurons are sort of working together to give you this temporal code on the bottom that really is locked again to the phase. And this is the total response. But each one can't lock exactly to the phase. It gets it every few times through, every few cycles. Okay, it's the Pauli principle. Okay, so we have place theory and periodicity theory are this Pauli principle, temporal theory. Okay, remember, place theory is based on the traveling wave. We talked about one problematic perceptual phenomenon, which is a missing fundamental. All right, so I have three frequencies at 1200, 1400, and 1600 hertz. What frequency is the pitch of the signal that I'm going to hear associated with?
200 hertz.
How much?
200.
200, right, 200 hertz, because the distance between these is 200 hertz. Okay, so if I play just these three components, I'm going to actually hear something that sounds like it's got the pitch of 200 hertz. That's the missing fundamental. There's no energy down there. Okay, and so the two theories can't equally predict that. So place theory would say that there must be some energy down there, and that's why I'm hearing that, right? So there must be some movement in the basilar membrane down there. So if I put a masking noise there, then I should not be able to hear it anymore, okay? But the fact that I can, right? There's no real energy there, okay? All right, let's move on. Just a little bit on this auditory pathway.
I have a question.
Yep, sorry.
So, do we need to know all the theories of pitch, like the points of agreement?
Yeah, so let's go back.
Or did you just want us to know place theory?
No, you need to know the temporal theories too, which are, that uses the phase locking and the Pauli
The phase
principle.
locking and
Right.
Pauli principle.
Yeah.
But
Okay. So
the-
one is based on timing and one is based on place.
Right, but the next slide, that, do you mean like this, like the points of agreement?
Well, so we know this anyway, right? Frequency is always the stimulus for pitch,
Yeah.
right? So we know that anyway. And we talked about Ohm's law a second ago, which is just basically Fourier analysis. Okay? So yes, you want to know that stuff, but you know that already anyway. So I wouldn't worry about that too much. And we do go in a little bit more detail about the missing fundamental in our full lecture, if you wanted to take a peek at that. All right. So again, auditory brain structures. So here are your cochlea. Signal goes to the cochlear nucleus on each side. Okay? So the signal on the right ear goes to the right cochlear nucleus, and the left ear goes to the left cochlear nucleus. Then we go up to the superior olive, and this is the first place where we get input from both ears, in the superior olive. We talked about two parts of that, the medial superior olive and the lateral superior olive. So there are two places. All right? But it's all the superior olive. It's the first place you get input from both ears, or binaural input. Okay? Then the inferior colliculus. Okay? And that's as much as we're going to need to know for that. Let's talk about psychoacoustics and these equal loudness contours. See if we can remember what that means. So this is our that threshold of hearing curve again. And we can see we're sort of maximally sensitive down here. All right? If I played 1,000 hertz tone at 60 decibels, okay, it's going to sort of sound like it's going to have, I'm going to rate it to have a loudness level. Loudness, again, is my psychological percept of this. Okay? Now, if I wanted to have a 100 hertz signal sound as loud as that 1,000 hertz signal, okay, if we follow this curve, I'm going to have to present it at more like something closer to 70 decibels. So what this is telling us basically is that the auditory system is not equally sensitive to all frequencies. Our pitch perception, our loudness, our perception of loudness is not the same for all frequencies. Okay, so if you look here, low frequencies have to be turned up a lot to sound as loud as the high frequencies. Well, sorry, there's these middle frequencies. Higher frequencies get a little squirrely there. But a lot of times you'll have a bass boost on stereo or something, or a loudness knob on stereo, or you might do it on, it might have that on your, well, whatever system you're listening on, okay? And that's what's happening there is they're actually boosting the loudness so that, whatever level you're actually playing the signal at is going to sound louder because you're getting the bass up. Because bass sounds not as loud for something presented at the same level as something in this sort of middle frequency region. Okay? Does that make sense, or am I making it more confusing? Okay. Well, you tell me to stop if it's getting too confusing. We talked about masking, all right? And if you're masking a tone with noise, you get maximum masking when the tone and noise are at the same frequency. Okay? If I took the masker and I was moving it, and I was looking at how loud that frequency needed to be in order to be detected, okay, as I got the masker closer and closer, I would have to turn up the level of the signal in order for it to be heard, that level of that target signal for it to be heard. Okay. We also see that these curves don't look symmetric, especially at higher levels. You get more masking out here, right, than you do down here. Here it looks like it's going up in a pretty, look like it could be, it would be a symmetric thing if it went straight back down, but we get more masking out here in these upper frequencies. This is the upward spread of masking. Okay? All right. Yeah?
What do you say about tone and noise?
Okay, so let's say I've got a masking stimulus right here. Okay? And I take my signal, I have a 100 hertz signal, and I have to see how loud I have to make it in order for it to be heard. Because the masker's all the way over here, I don't have to turn it up very much. But as I move that frequency closer and closer, okay, to where the masker is, I have to turn it up louder and louder to hear. Okay, so if things are the same frequency, they're going to be hard to hear. Okay? Then as I move it away again, I can turn it down more and more and still hear it. Right? Because the masking is concentrated in this frequency region. But I'm still getting more masking out here than I might predict if I were just using this side to see what I would get. So it's not symmetric. There's still additional masking out here, even at these higher frequencies. And that's the upward spread of masking. And that's what we talked about as being the basis for MP3 players. Okay? Masking. All right? This is a broadband stimulus. We talk about the low frequency cutoff and the high frequency cutoff, and we can vary it. And what we're trying to do now is see how much we're going to mask a signal, so how much we have to turn up a signal to be able to be heard in this noise. Okay? And we're going to vary the bandwidth. Okay, so consider a task in which you're asked to detect a pure tone embedded in the background noise. Okay? The noise is bandpass. That means we're only allowing a band of noise through. It's got that low and high frequency cutoff, and we're just varying that. And we're going to vary that bandwidth. Okay, so remember we look at this, so here's my target, it's that yellow line on the right.